You are now detectives investigating a murder mystery. The only clues are a strand of hair, the butt of a cigarette, and a single drop of blood. You can use these clues to find the killer because all of them contain DNA, which is unique to every individual. But what if I told you, although your suspects look different, they all have the same DNA? And that's because your suspects are different parts of the same human body. It's amazing if you think about it, how your eyes respond to light while your ears respond to sound if they both have the same genetic code. The answer is that every cell only uses a small part of the DNA, and the set of instructions that tell a cell which parts of the DNA to pass over and which parts to use are called epigenetics. I am studying the epigenetics of type 2 diabetes. In diabetes, the pancreas doesn't make enough insulin, leading to uncontrolled blood sugar levels. Left untreated, that puts patients at risk of heart disease, kidney failure, blindness, and stroke. So we know something is wrong with the pancreas. In particular, beta cells that are responsible for making insulin. But exactly what happens and why? The pancreas is a pretty complex organ. It has many cells, each with their own job. Acinar cells make digestive juices that are transported by duct cells to the gut. Beta cells make insulin, but they do not work alone. Could the beta cells have accomplices in diabetes? To figure out how the pancreas in people with diabetes is different from the pancreas in healthy people, my lab mates collected samples from human donors. The problem is these samples are heterogeneous. They contain many cells belonging to different cell types. If the composition of these samples is not constant, and I were to just sequence these cells and look at their epigenetics, then any difference that I would see would reflect that instead of telling me something about the disease. And in real life, it's not so easy to tell the different cells apart, especially when you're looking at their DNA, which is the same in every cell. This is the challenge that I'm trying to solve, and I will show you how epigenetic information can give me the solution. One type of epigenetic information is DNA methylation. It is a chemical modification on the DNA, and we can think of it like a fingerprint. It doesn't change the DNA sequence itself, but it can give us a hint as to where a DNA molecule came from. I wrote an algorithm to find unique fingerprints to the different pancreatic cell types in order to measure the composition of my heterogeneous samples. Now let me show you how this actually works. Can you guess which digits are used to unlock this door? You can do that because the use patterns tell you something about the importance of each digit to the code. Now, to do any job in the body, a cell has to make proteins, and the instructions on how to make proteins are written in the DNA. If a cell uses a piece of DNA a lot, then that piece doesn't get covered by methylation. But if a cell doesn't need a piece of DNA, like acinar cells don't need to know how to make insulin, then that piece gets covered by methylation. If we now sequence that cell and look at the methylation pattern, we can learn what that cell was doing. And if we know what the cell is doing, we know what cell type it is. Now, if we go back to my bowls of M&Ms, I can now estimate how many I have from each kind by just counting the unique fingerprints. This is interesting in itself. If, for example, in samples from diabetic individuals consistently have fewer beta cells, maybe those cells are dying, and that's why they're not making enough insulin. But more importantly, if we know exactly how many cells we have from each kind in each sample, then for any change we see, we can say if it is just due to a difference in composition, or if it could actually be associated with a disease. Now, I'm not the first person to use these unique fingerprints to count the different cell types in heterogeneous samples, but this field has changed a lot. New sequencing technology gives us more information on methylation than ever before. Looking at, the algorithms haven't kept up. Looking at the new sequencing with the old algorithms is like looking at a picture of this dog without 3D glasses. We get the information, we see it's a dog, just like we would have if this picture was taken with a regular camera. But there is an entire extra dimension of information that we're missing. My algorithm takes in all the information, like putting on 3D glasses. What happened was, in the past, we were only able to look at fingerprints in a few spots across the DNA. And now, with whole genome sequencing technology, not only can we look at many different spots, now single molecules can have several fingerprints on them. The old algorithms treat each fingerprint on its own, and my algorithm understands that a single molecule can only come from a single cell. I started by testing my algorithm against the old one on simulations. I made a make-believe pancreas with as many cell types as I want, in this case 10 different cell types. And I decide how many to put in from each cell type. And the levels that I decided are shown here in the red lines. Now, the closer the boxes are to the red line, the better the estimate. So on the old 2D technology, my algorithm and the old algorithm are exactly the same. But on the 3D technology, my algorithm does better because of the added dimension of information. To summarize, we saw how use patterns in the cell create unique fingerprints that we can then utilize to measure the composition of samples of the pancreas. Right now, I'm testing my model against the old one on mixtures of real data. I decide how many M&Ms to put in from each color, and I measure how well my algorithm can count them. The next step, once we know the proportions, is to figure out which cell types at which parts of the DNA are different in diabetes. If we know which cell types are changing, figure out what makes a cell diabetic, maybe we can slow down this process, stop it, or even reverse the damage, restoring pancreatic health. And with that, I thank you for listening and open for questions.
Thank you so much for the beautiful presentation. I'm wondering how similar this might be for type 1 diabetes. If you can relate to that as well. Thanks.
Okay, so there are two types of diabetes. And type 1 diabetes is the minority. It's only 10% of overall diabetes. And that is an autoimmune condition where the body attacks the beta cells and kills them. But in type 2 diabetes, as far as we know, at least not all the cells die, and we're not making an autoimmune attack against them. But they're not functioning as well as they should be. It's called pancreatic islet dysfunction, and we don't know exactly why that happens. So I think there's a lot to discover for type 1 diabetes, and we're still not close to a cure for type 1 diabetes. But the mechanism of how insulin is missing is better understood. Well, in type 2, it's still a mystery.
Hello, thank you. I'm Zivor Paz from Bar-Ilan University, biology. I want to ask you, besides the histone methylation, like you said, or other modification, maybe there's a connection to transcription factors. Like we know the transcription factors are related to the morphology of the cell and what is fate going to be. There is a difference, let's say, correlation between the some translation factor to the methylation of the histone. Maybe they change the morphology of the cell or something like this, the beta cell.
Okay. Well, thank you for that question, first of all. Here I'm not trying to make any mechanistic claims just yet. And I think DNA methylation can both be a cause and a result of changes in the cell. So for example, if you take an enzyme and remove a piece of methylation, you can cause gene expression by just taking away methylation. But the other way around, usually repressive complexes with many transcription factors first have to be present, repress a sequence, and then it is locked in by a DNA methylation pattern. So it's not the entire picture, but if we see regions that are activated in disease cells or are silenced in disease cells, that would be reflected in their methylation patterns. At least I hope so.
Okay, thank you very much.
Hi, Shachar from Tel Aviv University. Just a thought about if you see a pattern in the beta cells in type 2 diabetes, which we know that the beta cells, right, are not, the problem is not the lack of insulin in these patients. Could it be a response to the high level of insulin that is not being used? So like a form of trying to shut it down to mitigate the effects of insulin, because if you have insulin that's not being used, it can cause in itself different problems. So could it be like a way to?
I guess that two hallmarks of type 2 diabetes would be the pancreatic islet dysfunction that I'm investigating, but also peripheral insulin resistance, meaning the body is at first trying to make a lot of insulin to get sugar into cells and out of the blood, but the different tissues are ignoring that insulin signal. So at first in the disease, the insulin levels are very high, and then something changes in the pancreas and it kind of stops producing as much insulin as it did before. So it could be a result. It could be that the pancreas is responding to processes that are happening outside of it. But I think it would be interesting to understand anyway. And even if it is a result of a process that starts elsewhere, if we can stop the process at the pancreas level, and that stops diabetes from developing or from worsening, that's good enough for me.
Hi, I'm Sima. I'm here from the Hebrew University, molecular neurobiology. Okay, great talk. I wanted to ask about the algorithm that you're using. So I'm coming from the RNA field, and I know that there are many algorithms focused exactly on what you're doing in RNA, decomposing bulk samples to single cell. So do you think these algorithms could be also implied on DNA, or is it not possible because of fragmentation of the molecules? So your algorithm would be specific to DNA as opposed to the old algorithms.
Okay, thank you for that question. Right. There is a lot of interesting overlap between methylation and RNA sequencing. And in fact, there was a paper maybe a year or two ago that used RNA sequencing to do decomposition of methylation sequencing. So that those are both very interrelated. In DNA methylation, it's a binary code. There are only two states. Either a piece of DNA is or isn't methylated. So my algorithm is specific to that type of digital data, but it can, of course, be extended and expanded to include more states and could possibly be applicable to other scenarios.
Hi, I'm Hagit from education, but it's a great talk and very interesting. But I would like to ask about the potential for your algorithm to be relevant for other fields, other disease in the body. Is it specific for diabetic issues, or maybe it can be used and generalized to other disease and other type of cells and this ordering of our body?
Thank you. So my research is focused on diabetes and the pancreas, but the algorithm doesn't know that. So it can definitely be extended if we're talking about DNA methylation, any disease, any process in the body. And this is a really developing field. There are a lot of, for example, early discovery of cancer using DNA methylation, because the methylation is very different in cancer cells. So you can take maybe a blood sample and find some cancer methylated DNA within it, even though the DNA is very similar to the DNA in the rest of the body. So definitely has many applications. As far as completely different fields, it's funny because my husband is doing research and he is studying geography, and he's looking at the satellite images of Israel and is trying to figure out where plants are, where there are trees and where there are bushes, and I'm sure I'm butchering his research. But we find ourselves tackling many of the same issues because he's trying to measure how many, what is the proportion of trees in this field, and I'm trying to measure the proportions of beta cells in this field of the pancreas. So the ideas are very broad and similar and relevant to different fields.
Anyone else? Thank you very much to Irene.